Good afternoon, everyone. May I have your attention? All right. Last Jones seminar of the term, so bear with me. Also happens to be the last of this academic year. I'm sure there are some who are quite happy about that. All right. Today's speaker is Dr. Kadoury from the École Polytechnique de Montréal. Dr. Kadoury is an assistant professor in the Computer and Software Engineering Department at Polytechnique of Montreal, member of the Biomedical Engineering Institute at the University of Montreal, and researcher at the Saint-Justine Hospital Research Center. He currently holds the Canada Research Chair in Medical Imaging and Assisted Interventions at the Polytechnique of Montreal. He obtained his bachelor's in computer engineering in 2002 from the Polytechnique of Montreal and his master's in electrical engineering from McGill University in 2005. He completed a postdoctoral fellowship at the École Centrale in Paris, and worked as a clinical research scientist for Philips Research North America and the National Institutes of Health in Bethesda, Maryland, from 2010 to 2012. Professor Kadoury has published over 60 peer-reviewed papers in leading journals and conferences in fields such as biomedical imaging, computer vision, radiology, and neuroimaging. Please welcome our speaker.
Can you hear me? Yeah? Okay. Thank you very much, Alex, for the presentation and for inviting me today for this talk. I'm quite happy to be here. So this afternoon, what I'll be giving as a presentation is kind of our core research in our lab at Polytechnique, which revolves around image-guided interventions. And the topic of the talk is trying to explain on the different types of technology we're developing in medical computer vision to for the needs of image-guided interventions. So what I mean by that, I'll try to explain it with this graph, is if we look at the different steps that a patient goes through for an image-guided intervention. So I know there's a lot of students here who work in the medical field in engineering. For those who don't know a bit about it, it's really just trying to assist the surgeon or an interventionist with medical images to guide the surgeon or the interventionist to actually reach a target or treat a tumor with a certain therapeutical device. And if we look at the different stages, there's the diagnostic stage where there's actually visualizing a lesion or a tumor with diagnostic imaging. Then the patient goes through the reaching phase where the surgeon or the interventionist is trying to reach for a target. So that's the actual guidance step. The last step is the treatment phase. So this is where there's a monitoring of the therapeutical procedure and try to assess the success of the therapy. And if need be, the patient goes through a cycle. We hope not. But if there's a recurrence of a tumor, then there's new diagnostic imaging. So in all of these three principal steps, there's some key medical imaging techniques or image processing techniques that come into play that try to ease the workflow for the interventionist to try to get multimodal fusion or being able to segment certain structures of interest from these images. And that's the kind of the technologies I'll try to talk about this afternoon. So obviously I'm not alone here. I'm surrounded by a great team at Polytechnique and also a few number of academic, health, and industrial partners that makes all this possible. And it's really this tri-party collaboration that allows for a transition of these technologies into clinical practice and maybe also in a commercial product. So we have a group of radiologists, interventionists, but also engineers that work on the different projects. So the focus of the talk will be on image-guided interventions. I just put this slide on what we're also working on in our lab. We're working on neuroimaging for specific diseases like epilepsy or spinal cord atrophy, multiple sclerosis. We also work on spinal pathologies. So we develop some techniques for spine segmentation and multimodal spine fusion. And we also work on brain tumor segmentation, which actually is a project which we initiated with Pablo Valdez on some of this for kind of being able to plan an interventional neurosurgery. So the perspective I'm going to try to give here is trying to solve some of the problems that a radiologist and an interventional radiologist is faced with on a daily basis. If we look at the radiologist perspective, he's faced with on an increasing amount of work on a day-to-day basis. More and more cases he needs to treat more and more cases every day, and difficult cases actually increase the workflow time. Try to get comparable cases to give the proper diagnosis. So there's a workflow inefficiency in that case that's we're trying to solve. Also, in terms of the interventional radiologist, there is some time lost in trying to localize some superficial lesions if we're talking about cancer patients. Some lesions that were identified by radiologists, but needs to be re-identified in some interventional imaging from the interventional radiologist. There's some inefficient workflow there that could be improved. Also, the interventional radiologist will try to handle complex targeting, so some try to reach some targets that are difficult to attain, avoid some critical structures during the intervention, and it can impact patient care and interrupt the workflow. So it may affect the quality of the outcome. And what we're looking for is really try to bridge the gap between the work from the radiologist and translate that into the interventional setting, to try to fuse information that was planned before the procedure in the operating room or in the interventional room. So the first clinical application I'd like to talk about is on liver oncology. So in liver oncology, we could potentially try to divide that into two different worlds with two different solutions. In the Western Hemisphere, in North America and in Europe, we're talking mostly about metastatic liver cancer, where the primary cancer is colorectal or pancreatic cancer. You will see metastases appear in the liver. So it could be one to multiple metastases of different sizes. The second part, maybe more in the Eastern world, in the emerging markets, we're talking about mostly a primary liver cancer, such as hepatocellular carcinoma, which is a primary liver cancer pathology, and with over 1 million new cases diagnosed every year. And if we look at some typical images of metastases and HCCs in cancer patients, this is kind of what it looks like. So they're really different pathologies. And the first clinical application I'll present today is really more on treating metastases for liver. So one therapeutical solution to treat this, or to eliminate metastases, consists of radiofrequency ablation in the liver. So this therapeutical procedure consists of guiding a radiofrequency needle through the intercostal ribs and guided to a tumor inside of the liver. So once it reaches a tumor, this needle would create a radiofrequency, so it would create some heat and create a coagulation of the blood in the tumor, creating therefore necrosis of the tumor. And depending on the size of the tumor, you may require one or multiple ablation zones to actually cover the entirety of the tumor. And this procedure could also be repeated if there's multiple metastases inside the liver. So RFA, or for radiofrequency ablation, it's a minimally invasive procedure. So it's usually image-guided, either ultrasound, CT, or MR. And it's an FDA-approved soft tissue ablation procedure. It can also be applied not only for the liver, but also for lungs, kidneys, or bones. And is applicable for patients not susceptible for resection. So for these cases that are not needed for resection, will be approved for an RFA. So when we talk about image guidance with ultrasound or CT or MR, if we take each of these modalities independently, each come with their own set of limitations. If we look at ultrasound, it's 2D or starting to be 3D, but it's still very low or no targeting the contrast unless there's an injection of contrast agent. Imaging access is quite difficult between the ribs. So as you've seen in the animation just before, the needle has to be guided through intercostal ribs. So there's some challenge to actually guide that to find the correct entry spot. If we look at CT, it's still intermittent. We need contrast agent to have good contrast between the tumor and the liver. And MR, although it's starting to be more accessible, is still costly, and access to the MR is still of a challenge. I'm going to talk a bit about solutions of in-gantry MR guidance, but still, if we look at the reality today, it's still quite a challenge to have real-time MR guidance. So to actually plan this, so what we're looking for is really to have a fusion system that will be able to combine multiple modalities where we have a diagnostic on the CT or an MR and fuse that to intraoperative ultrasound. So if we look at the planning stage for procedures, so this could be done the day before, just before a patient goes into the interventional room. The interventional radiologist will identify the tumor, also plan the number of ablations required to cover the entirety of the tumor and the skin entry point. So I'm not going to go through all of the detail in terms of the planning. There's a lot of actually research going on to actually optimize the planning procedure to find the best spot to actually insert the needle and how to go about to reach the target. There's two key technologies that we're working on that is of use for the registration problem, but also for clinical aspects. So the first one is on liver shape segmentation. So this will allow us to get some information on the volume of the liver. Will also help for the planning stage for the skin entry point. If we have a segmentation of the liver, we could have more detailed information on where to actually insert the needle, and also allows for PET-CT fusion. Again, I'll show a bit more detail on that later on. But it's really a key stepping stone to the registration procedure for the liver segmentation. Also, another technology we're working on is actually tumor segmentation, which could give an estimation of the ablated zones and automatically give an estimate on the number of ablations to actually cover the entire tumor. It's also of good value for post-procedural assessment to be able to see if there's recurrence of the tumor or if the ablation actually covered the entirety of the tumor, if there's any recurrence. It's a good tool to actually be able to quantify if there's any residual tumor after the procedure. So one of the technologies we developed is a CT liver segmentation technique, which will really be able to delimitate the boundaries of the liver from a CT image. So the technique, you'll see a lot of workflow diagrams. As I'm an engineer, I like to put a lot of workflow diagrams. So that's one of the, to actually show how the liver segmentation technique works for a CT. So we detect the skin and approximate the position of the rib cage and detect the position of the lung and diaphragm. So this will create a seed point inside the liver and will propagate a mesh inside the liver until it finds the optimal boundaries. So basically, we minimize a cost function which will regularize the topology of the liver and attract the boundaries of the liver to the highest gradient in the image. So that's really the driving force that will deform our initial mesh. And the third term is more of an intensity-based, region-based term, which will then on-the-fly compute statistics on the distribution of the intensities inside of the liver and try to keep the same type of distribution as it's propagating towards the edges. So this will allow to actually have an homogeneous region inside of our segmentation. So it's quite a quick approach. It usually takes between 25 and 30 seconds to get a segmentation in a supervised manner. So we validated the approach with radiologist segmentations on 20 crown tooth data sets. And we compared that to different approaches, only a voxel-based approach or a landmark-based approach. So the intensity-based approach is what we actually proposed. And it gives us quite good numbers in terms of mean surface distance and also dice coefficients. So that's the intersection between the ground truth segmentation and our own segmentation that was generated automatically. So I'm giving some images here, some sample results that we had. Obviously, this needs to be applied to pathological cases. So just applying it on normal liver, there's not a lot of use into that. This is really to be used for patients with cancer and that shows different metastases or different ablated zone. So one of the problems actually that we saw with that technique is this was actually a case that had a prior ablation. The segmentation didn't really include that in the segmentation. It's up to really to the radiologist to determine if this needs to be included or not. But that zone really showed an intensity distribution which was understandably quite different than the normal liver tissue in that region. So in that case, it really didn't include the prior ablation zone in the liver. So that's one problem that we're actually looking into to try to take that under consideration during the segmentation process. If we look at the MR problem, so we have CT segmentation. It's only an intensity-based procedure. We usually have good resolution for those type of images. For MR, it's quite different. For abdominal scans, usually there's maybe 8 millimeters in space in between slices. During the length of the acquisition is quite longer as well, as you know. So we need to, in that case, we can't really assume only an image-based approach. We need to incorporate statistical information to be able to segment the liver from the MR. So in that case, what we did was we created a database of training cases from prior cases. So we had manual segmentations where we would delimitate the boundaries of the liver and then create a statistical shape model. So here we incorporated statistical knowledge of the appearance of the liver for segmentation purposes. So we built the standard SSM model with the principal components that actually show the principal modes of variation of appearance in the liver. So this helps us to generate a triangulated mesh model, which we use to actually adapt if we're giving a new MR to actually segment the liver automatically from the image. What we're bringing as a novelty to this approach is that for each vertex in our a priori model, we assign feature functions to determine what's the optimal boundary when it's actually optimizing that model. So it's quite a lengthy process during the training phase. So it maybe take around 20 hours to actually take all of the cases and test all the different possible configurations when we're trying to displace these vertices in the image and find what are the intensity distribution for all of these positions. So we test various configurations and try to find the optimal distribution that will give the optimal boundaries from the training set. So we train some feature functions based on this training data set, and we find the optimal boundaries, optimal ranges, which will find these features which would give the precise boundaries on the MR image. So we train this offline, and once we have a new case coming in, we input that MR image into the process, and it will launch the formal adaptation of that mesh, trying to minimize a cost function which will try to attract the mesh to the optimal boundary. So this is the search strategy that we adopt. If we have, for example, a vertex here and a vertex there, the search direction will go along the gradient of the image. So if we have a normal on our triangle here, the search direction will go along the gradient of the anatomical object. And the internal energy will try to ensure some consistency in the mean model, meaning that we'll try to still follow the mean principal modes of variation, but still try to get an end result that is as close as possible to the normal variations of the appearance of the liver. So we studied the accuracy of this approach on 30 T1 weighted MR scans with a 70-second delay in post-contrast injection. Why that specific sequence is that it's lengthy enough to actually let the contrast agent propagate throughout the liver and give the best contrast in comparison to the actual background. So by discussing with our radiologists, we found that was the optimal sequence that would be used for the training and the testing. And you see here kind of the challenge is really an 8 millimeter slice spacing, which is quite a constraint to actually try to have a purely image-based approach, and why we needed to incorporate statistical knowledge in our framework. As for each case, we had 10 data sets which we evaluated the errors and computed the mean distance and so on. So again, these are some sample results on 4 cases, and so this is completely unsupervised. So really, it will find the initial point to initialize the mean model and adapt that to the boundaries on the image. And the few challenges that we could see here is that we did a first training on maybe 10 data sets, and what we found is that it wasn't enough to actually learn the best delimitation between the kidneys and the liver. What we saw is that in a lot of cases, actually, it would include the kidneys inside of the liver, because the transition and the boundaries is quite similar between the kidneys and the background and the liver and the background here. So we really need to increase the size of the data set, and that's really we're seeing a trend in medical imaging with a lot of machine learning techniques starting to be used, and we really need to have a well-represented data set to show the different types of liver appearances and different contrast levels, because each patient absorbs the contrast in different fashion. So we really need to have a good representative distribution of the images. And here, if we see the distribution of errors, in the majority, they will fall between 0 and 1 millimeter. The greatest challenge is really on the top of the liver, where because of respiratory motion and because of the poor between slice spacing, will give some interpolation error in this case. So that's where we see some of those challenges, but overall it gives quite an accurate segmentation. So if you look at the different numbers, it gives some, if we compare it between fatty livers and normal livers, it actually gives slightly better yet non-significant differences between large livers and low-margin livers, but in mean difference would give 1.9 millimeters, and would fare fairly well compared to state-of-the-art methods, for example, compared to point-distributed models or level set approaches in terms of state-of-the-art. So that's for the liver segmentation challenge, liver segmentation problem, and I'll show how we actually use that in the registration problem intraoperatively. Another problem we're looking into is the tumor segmentation, which is very important for the planning stage, but also, as I said, for the post-operative assessment of a procedure. So two different approaches we're looking into. First is a more of an interactive approach, so kind of give the radiologist, keep them in the loop during the segmentation process, but it really eases work to actually segment a tumor. So here we're using an interactive approach. Instead of using Euclidean distances, we're using radial basis functions to actually help for the segmentation process. And we're using different features such as the gray level thresholding, simple edge detection, or texture analysis to actually find the best boundaries between the liver and the tumor. One of my students is actually looking to a purely unsupervised segmentation approach that's adaptable for CT or MR. And here he's looking into texture analysis using a gray level concurrence matrix. So for example, using energy, entropy, kurtosis, variance measures to actually learn how the texture differs between the tumor and the normal liver tissue. And this will give an initial segmentation map and then adapt a non-omnidirectional deformable model to find the best boundaries for that tumor. So that gives some initial results for an NMR scan. So it gives some. So it's an ongoing work right now, but really looking into more machine learning techniques to try to learn of the best way to actually discriminate normal from pathological tissue. So that kind of looks at the different technologies we're developing for diagnostic imaging. If we're looking at the second phase, so if you look at my first chart, there was 3 different phases. The second phase is really the reaching part, which I'm sure a lot of you are already familiar with, where we're trying to fuse information that was identified from the radiologist, so tumor segmentations or structure lesions we're trying to biopsy, or critical structure we're trying to avoid, and bring all that information in the interventional radiology. So being able to fuse preoperative imaging to intraoperative modalities. So really the objective is trying to assist the physician in the execution of procedures with target inclusion. Why we're trying to do this is trying to, for example, minimize damage to critical structures and have a quicker localizations of the target we're trying to reach. So being lesions or anatomical landmarks. So really try to streamline the workflow and be able to find targets more accurately and in a faster fashion. So all of this is embedded in the minimally invasive treatment paradigm, where there's no blood loss, no stitches or scalpels or scars. So the secondary effects are fairly reduced. And usually patients are admitted for only one night. So they come in, get their treatment, they stay overnight, and then they're released, discharged the next day, since it's not an invasive open surgery. And the key message here we're trying to we're trying to make understand is that there's no single image modality that is used independently. Really, the key here is the complementarity between all these different modalities and brought together to have a comprehensive image that will be given to the surgeon or to the interventional radiologist. So it's really the complement between metabolical, functional, and anatomical imaging that will be able to really have a complete picture during the intervention. And we're trying not to lose all this high-resolution information that was taken from the diagnostic images and have that available for the surgeon during the procedure. So it offers quite an efficient medium to actually visualize all the different functions or the morphological information during a procedure. What I've been working on a lot in the past few years is mostly on electromagnetic tracking. So what we're trying to have here during intervention is being able to track instruments or probes during an intervention in real time. So knowing where they are in space and know how they correspond to, for example, a CT or prior MR image. I know in neurosurgery there's a lot of work done in optical imaging. For oncological procedures, we need to track these devices inside of the body with, for example, a biopsy needle or even catheters. We want them to have the precise position and orientation during the intervention. So the problem here, or the challenge is the way that it's blending, but if we're given a pixel in an ultrasound image, we want to have its corresponding pixel on the CT. Basically, that's what we're trying to achieve. So, for example, if we have a target that's identified as the CT, we want to know during the procedure on the ultrasound image where that tumor is. So to know where that ultrasound probe or where that needle is in space, we use small coils, which I think the image are just here. So these are the small coils that we use that we embed either in the needle or that are fixed next to the ultrasound probe. So at a 30 hertz frequency, we're able to get the real-time position and orientation of these tools inside of the body, even if it's not line of sight. So even if it's deep inside the liver, we're able to get that information in real time. And we need a registration procedure to have that correspondence between ultrasound and CT. So here I show the chain of transformations that bring a pixel in the ultrasound image to the CT coordinate system. So these two transformations are known. Here we do a prior calibration to have the mapping from a 2D pixel to the coil, the small coil. And that small coil actually, when they're under a very, this is a field generator, so it generates a very weak electromagnetic field. And the current that passes through the coil gives us the position of that sensor. And that transformation is given in real time, so that's the transformation between the coil and the generator. What changes every time a new patient comes in is the transformation between the generator and the CT scan. That's what we need to determine with an automatic registration. A lot of techniques right now in the commercial system will use a point-based manual segment registration approach. So what we're developing is trying to automize that framework to be able to streamline the first phase, which could, in practice, take maybe 10, 15 minutes. If it's with a point-based, or if really going manual, it could take a lot longer than that. So we're trying to really automize that framework. So that small coil that I just talked about, we can embed that in thermocouples, in biopsy needles. And the system we're using is the Aurora system from NDI. And this is a picture of how we set up the generator on top of a patient. Here we see some small markers, which are the manual markers we use for manual registration, and we compare the automatic registration with these point-based, fiducial-based registration. So here we're simplifying the problem in order to have an image-to-image registration problem instead of doing point-based registration. So we'll use the ultrasound imaging that was taken intraoperatively to be able to perform this registration. There's two different types of 3D ultrasound. There's two different ways to actually generate 3D ultrasound. The first one is using 2D tracked ultrasound. So since our probe is tracked in space and in time, then if we do a liver sweep, as it's shown in this video here, we ask the interventional radiologist to just place the probe and sweep through the liver as much as possible to create a 3D volume of the liver. So by taking, it's like 2.5D images that we get, and by interpolation, we're able to create a volume in 3D. That's one solution. Another solution is more and more we're starting to introduce three-dimensional probes. So one of those probes is the X61 by Philips that we were using to actually get instantaneously a 3D volume of the liver. You could already see the difference here. This liver sweep could take maybe 8 to 10 seconds. So if we have a patient that has a biopsy, a free breathing biopsy, he's not under sedation, the interventional radiologist will ask the patient to hold his breath, which is not necessarily guaranteed. So when we do a sweep, we might see some artifacts which kind of brings in some errors in the 3D reconstruction. If we compare that to the 3D probe, in a fraction of a second, we're able to get that 3D volume and avoid these artifacts due to breathing motion during the reconstruction process. So again, another workflow diagram to actually show how we go about the registration problem automatically is we take that 3D liver segmentation from the diagnostic images that's shown here. So we get a triangulated mesh model. And from the ultrasound volume, we extract an inner surface, and we'll try to match both of these surface models together to have a registration between the ultrasound and the CT. Since the ultrasound is tracked, then we have a correspondence between the tracking space and the CT space. And we also use the skin surface of the patient. Why would you actually use that is we'll use that as a constraint during the optimization process. Since we know physically that the probe is sticked on the skin of the patient, then when we actually have the registration, we'll be able to check if it's the solution that it's giving to us actually maps the position of the probe onto the skin. If, for example, if it gives us a 4x4 transformation matrix that brings the probe here, it makes no sense. So we'll highly penalize that configuration. So as an output, what that will give us is a fused CT to ultrasound image, which will enable the interventional radiologist to actually find a tumor and be able to reach that with the ultrasound imaging. Just a quick word on how we actually get that surface segmentation from ultrasound. We initialize a seed inside of the 3D volume that falls into near the diaphragm and apply a dynamic region growing to be able to extract that region and apply a ray tracing algorithm to extract the inner surface of the diaphragm. Then we expand that surface using a graft brace approach. I'll explain just in a second why we need that expansion. Here we have some results on that surface extraction from ultrasound, and you see a dropping signal from the ultrasound because either rib shadowing or there's another artifact that conflicts with the propagation of the ultrasound beams to that spot. So because of that drop of signal, the propagation actually stopped at that point. So we perform a localized search to actually continue the propagation of the surface and increase the size of the diaphragm extraction. That will significantly help for the registration process since we have more information to actually include in the optimization procedure. So that's, again, our cost function that we're trying to minimize. It's very similar to an iterative closest point, so it's an ICP algorithm. We just include regional terms to ensure that we have regional consistency during an optimization, and that's the penalizing term with the skin surface. So we evaluated our technique prospectively on 29 patients on 4 different clinical sites. 12 had liver RFA and 17 had biopsy. Why we actually categorize between these two is actually we'll see some difference in the numbers in terms of accuracy because of the breathing motion between RFA and biopsy. For RFA, they're sedated, so they're under a ventilator, and we can control the respiration during the procedure. While in biopsy, in some cases, they're free breathing biopsy, which will kind of bring some problems during the registration step. And we use both 2D and 3D probes to actually compare the efficiency of using 3D dimensional probes for the registration. So here we're showing some side-by-side images of ultrasound and CT. And just from the first one, you could really see the corresponding ultrasound. This is what the interventional radiologist sees with the ultrasound system, and the corresponding CT slice from after the registration. So here you see fairly well the pleural and the hepatic veins that comes up on this CT, which is not very visible on the ultrasound. And we have these results for CT and also for MR. So the interesting thing with this framework is that it's compatible either for MR or CT. Since we're treating a mesh model, we're just extracting information which is common between both modalities to register that with ultrasound. So here we're going through an axial and coronal view of corresponding, sorry, there's some glitches here. In color, it's the ultrasound image, and in background, you see the MR. So you see the boundaries fall quite nicely on the edges of the liver, and the vessels here, you have a good correspondence between the vessels, between ultrasound and MRI. So this is a clinical case with a free-breathing biopsy, where we have the corresponding meshes fused together and side-by-side images between tracked ultrasound and individual overlaid MR. And in that case, we had both prior CT and MR, so we could actually have a triple modality comparison to be sure that we have a good alignment with the first bifurcation of the hepatic tree. So we need still to quantify how good the registration procedure is. So we had a radiologist select some landmarks on both ultrasound and CT and compare target and fiducial registration error. And we were able to compare also with a purely manual approach. So we asked a technician before the procedure to actually do the whole procedure manually. In terms of accuracy, we're not as good as manual registration. They'll spend a lot of time to be sure that everything is perfectly accurate, and it actually shows with the time. So on average, 600 seconds to actually do the registration. So it takes 10 minutes to actually have a proper registration, while with an automatic approach, you could have that with 44 seconds on average. So it saves you time. The question is if the time that you're saving, is it worth with the accuracy that you're losing? So here you're losing maybe 0.6 millimeters, which in some cases is sufficient enough to go about the procedure. What's interesting also is that if we separate the results between 2D sweeps and 3D ultrasound, here we see almost a drop of, not twice, but we have a 2.76 TRE error in 3D ultrasound compared to 4.17 in 2D sweeps. So really you introduce a big error when you actually introduce that prior procedure to ask the interventional radiologist to do a sweep that will significantly increase the error of registration. So that framework was actually used in a study that we published in radiology, which was to see how we could fuse PET imaging for targeting of suspicious lesions using electromagnetic tracking. So here we're not able to just fuse prior CT or an MRI, but also fuse metabolical imaging during the intervention. On the left side, this is a case with cervical biopsy from a 71-year-old who had a B-cell chronic lymphatic leukemia with an avid FDG focus. So the lesions were actually not visible neither on CT or MRI, but they were visible on PET. And with this procedure, we are able to identify the targets on the PET imaging and fuse that to a live ultrasound. So the interventional radiologist was able to target these lesions with targets identified solely from PET imaging. So the procedure that we used is it was a PET CT, so we used the CT component of the PET to co-register that with the intraoperative imaging. And maybe I'll start a parenthesis, but really on the matching problem, and I'll revert maybe to some work we're doing in neuroimaging. The correspondences between the surfaces is not a trivial problem, as you know. Being able to map two surfaces together. The question is, how do we find corresponding points between our surfaces? If we look at the cortical surface, doing this comparison in Euclidean space is fairly challenging. So what we're looking into is actually transforming the Euclidean space into spectral domains. So instead of representing our shape in the Euclidean space, we'll do a transform to represent that in the spectral domain, and using the different eigenmodes to actually being able to match different shapes, for example, between one or two patients or between the different meshes that we have between CT and ultrasound. So this comes from some work from our collaborator that, in computer vision that, for example, if we have two different meshes, it's simply determining the Laplacian graph for each of these two meshes and doing the eigen decomposition of these Laplacian graphs to be able to extract the different eigenmodes for each of these meshes. So the matching process will not be done on 3D coordinates, but really on the eigenvalues from this eigen decomposition. So we're able to have some matching between our shapes. And so once we have a one-to-one correspondence, then that will enable us to have a diffeomorphic registration. So we're able to have an elastic warping between our diagnostic and the ultrasound imaging and compensate for respiratory deformation. And actually, we're in the process of transitioning to intraoperative registration. But first, we looked into a clinical problem that we're looking into for. It's different from what I'm presenting, but in terms of benign epilepsy, try to predict if there's any morphological changes between epileptic children and controls. And we actually use this framework where here for giving a normal with some segmentation and a child with a left foci in benign epilepsy, try to see if there's any morphological changes in some of these subcortical structures. So what we found is that using this matching method, we're able to detect some morphological changes in the left putamen for that patient compared to, for example, controls when we compare that to a normal control group. Which is we're not able to do with, for example, a standard ICP method, for example. If we really need that one-to-one correspondence to evaluate these morphological changes, and we're in the process of trying to adapt that for a real-time setting. The last phase I wanted to talk about is really the monitoring part. So once we're at the target, the interventional radiologist proceeds to the ablation. The limitations of image-guided ablation is that there's a high recurrence due to either underablation or inadequate tumor margins. That means there's not the appropriate heat that's applied to actually cause necrosis for a specific tumor. Obviously, each patient is different, so the amount of heat that has to be generated will fluctuate from one patient to the other. So one research question that we ask is that if it's possible to have intraprocedural monitoring during the procedure and not wait a few days after that the patient has a contrast-enhanced CT scan, we see if there's still presence of tumor and the patient has to go back. If there's a way that we can actually show a thermal map during the procedure so that shows actually a picture to the interventional radiologist if there's accurate coverage or sufficient coverage of a tumor during the procedure. So this will actually help to evaluate safety margins, or for example, if it's a composite ablation, being sure that the entire tumor is covered with the appropriate heat zones that is applied to the tumor. So our rationale behind this is that physiological changes occur during tissue heating. So as you increase the temperature inside of the liver, this will cause a dehydration of the tissue, causing a stiffening in these regions. And this will actually cause this bubble formation necrosis that's actually visible on ultrasound. Because the tissue is more stiff, the ultrasound sound waves don't propagate as good as normal tissue because there's no more water to actually propagate the ultrasound beams. So our hypothesis is that there's something that's going on in the imaging that's not necessarily detectable with the human eye. And we actually wanted to test this on either CT and cone beam CT to see if there's any changes that could be detectable during the procedure. So to create this thermometry technique, we did a calibration with a water phantom where we increased the temperature and got a cone beam CT to get the Hounsfield units for all of these temperatures. So once we had the calibration, our workflow to actually have a monitoring of the thermal map during the procedure is if we have a baseline image, so once the needle is guided, but we haven't started yet the ablation, we get a cone beam CT, and once the ablation has started, we get a confirmation scan and compensate for motion using a deformable motion compensation algorithm to realign our images. So once they're realigned, we apply an image processing technique, which is called the Röntgen filter. It's basically a first derivative filtering, which detects the changes between consecutive scans. So if you give one image and another image, it looks in a certain neighborhood to detect any changes in the Hounsfield units. And that unit will actually make that correspond to a certain temperature in our calibration map. So this is determined before procedure or with a water phantom, and we're able to apply changes in Hounsfield units to actual temperature changes. So we did two types of experiments to validate this method. We first did a swine shoulder experiment where we had a thermocouple and an ablation needle, and we compared the actual temperature at the tissue location and the one that is predicted with the thermal map. So here we saw with 3 trials, we have differences between 1, 2, or 3 centigrads between what is the result and the ground truth. So we have a pretty accurate prediction of how the heat is actually propagated throughout that region. So these are the temperatures for the ablation needle and the one with the thermocouple. So this kind of validated that we were on to something here. And we started to do some retrospective analysis where we actually had thermocouples, and we were able to compare the actual thermometry maps with the actual readings that we could get during the interventions. And in this case, it was, I think it was 79 degrees was the temperature measured at the tip of the probe, and 80 degrees was the predictive temperature from this thermometry technique. So this could actually be an opener for being able to monitor these procedures and correct for any discrepancies. For example, if there's multiple ablations, to have an appropriate coverage of the tumor before the patient actually is discharged and leaves the hospital. So I talked about electromagnetic tracking. We're also looking into optical shape sensing for tracking devices. If you saw in the picture, we set up this field generator on top of the patient. It takes some time to actually set that up, and it's kind of in the way of the interventional radiologist when he's trying to guide some needles. So in collaboration with our physics department, we're developing some probes with optical fibers, which integrate 3 fiber gradient sensors into the needle to be able to reconstruct a shape of a medical instrument during a procedure. So here it's a proof of concept on actual needle. And these are 3 optical fibers that are configured in a helicoidal fashion. And we measure the strains, which we use to actually reconstruct the shape of the needle in real time. And we actually also tested on sample catheters to be able to reconstruct that shape in 3D. So why we want to use that is I talked about metastatic liver tumor cancer. We're actually looking to develop that for guidance of TACE procedures, so for transcatheter arterial chemoembolization, where there's a pre-procedural planning where we need to figure out what are the feeding vessels for the HCC tumor that is shown here, and have tracking technology that's able to follow the position and the shape of the catheter as it's guided towards the feeding vessel. So this is an ongoing project in collaboration with Fred Leblanc, who was here, to actually develop the technology to do the registration and perform optical shape sensing to reach a certain level and track the position of the feeding vessels. So to actually achieve that, we're looking into different models to segment the vasculature. I had a student that's just published his method on extracting the spinal cord. So these are tubular structures that we could segment from MRI. So we're adapting that method to actually segment the vasculature inside of the liver. So these are, he calls it propagated deformal models. So it actually starts from the seeding point and propagates throughout the vessels and extracts the vasculature in an unsupervised fashion. So this is one of the ongoing projects we're looking into right now. So with the time that's left, I'll talk about another clinical application which I worked a lot on. It's in prostate cancer. It's pretty much the same formula as I showed for liver cancer, but here we have clinical results that actually showed the value of fusion imaging compared to standard of care ultrasound imaging or just MRI. So as some of you know, prostate cancer affects 1 out of 6 men in their lifetime, and in total in the U.S. around $8 billion in total expenditures per year. So you probably know someone in your family that has or had prostate cancer in his life. The problem is that there's still no gold standard imaging for prostate. Although MRI is starting to be a gold standard for diagnostic imaging, for real-time imaging, there's still an ongoing field where until now, transrectal ultrasound is still the modality of choice for getting biopsy inside of the prostate. Basically, trust ultrasound guidance is a blind procedure. It's basically just poking 12 spots inside the prostate and hoping you'll find cancer in the prostate. So it's pretty a random procedure. And the consequences of that is that this will actually cause between 50 and 30% of false negatives using standard of care trust biopsies. This will cause wrong diagnosis, delayed therapy, and anxiety for the patient. Also, because it's quite a global Actually, because it's more of a random fashion, therapy is still very global, meaning that it's whole gland procedures, either prostatectomy or whole gland radiotherapy. And the side effects are quite devastating, so either incontinence or repentance in between 30 and 80% of the cases, and can have a significant impact on the stress and life of the patient. And back in 2009, there was a paper in New England Journal of Medicine showing that to prevent one cancer, prostate cancer death, an additional 48 men had to be treated to actually to be able to save one life. So there's definitely some overtreatment that is done. It's not yet very focalized in terms of biopsy or treatment. So it's the same approach, but it's the same message we're trying to convey here, is that if we have information from diagnostic high-resolution imaging, from, for example, here, multiparametric MRI, we want to fuse that to intraoperative transrectal ultrasound. So we've developed a framework that takes the work, that's the identification of the lesions from the radiologist, and once the patient is either in the operating room and in the urologist office, we get a 3D representation of the prostate, register that with a similar technique that I presented for the liver, and now we could have a fuse MRI to transrectal ultrasound image fusion to actually target the lesions in real time. It's the same coil pretty much that we're using here. Instead, we're embedding that to the needle guide, to the gun that actually is used for the biopsy, and that's clamped on top of the transrectal ultrasound probe. For prostate interventions, the patient is still actually conscious. He's actually looking into our research platform during the procedure. So as soon as there's a biopsy taken by the urologist, obviously he'll react to that, and the registration might shift a bit compared to what we had initially. So motion compensation was a key issue here that we need to address. So to kind of solve that issue, we took some of the last ultrasound frames to co-register that with the initial 3D ultrasound. So that would help us to actually correct for any motion. So here you see that in green is the segmentation from the MRI, and you see in the background, the transrectal ultrasound. You see a discrepancy in the registration. So it's manually triggered. So once the user sees a discrepancy, it's co-registered again by taking the last few ultrasound images. So the clinical study here was on over 400 patients, where we used both our research and the commercial prototype. And the inclusion criteria was for PSAs that were higher than 2.5. So that's a prostate-specific antigen that's used to detect or follow the progression of potential prostate cancer. So two radiologists actually identified lesions on the images and were integrated into the Fusion platform. So we were able to evaluate that on 203 cases. And the procedures is that we would both do standard of care, transrectal ultrasound, and followed by the fusion system that would try to reach the targets identified by the radiologist. So we could actually compare the standard of care technique with this new fusion system. So we look at all the cases for the 193 cases that were able to be evaluated. If we look at the absolute number of positive biopsies, we don't see too much difference in the moderate or high suspicion. If we combine moderate and high suspicion, then we see a significant increase. In blue is the Sexton biopsies, so that's the standard of care. So we see an increase in absolute number of biopsies. The problem is that this is really the total number of biopsies, so it's not really representative of really the actual results for each patient. However, if we look at the positive biopsy rate per patient, then we really see a significant increase based on the MRI suspicion. These categories, low, moderate, high, that's really what the radiologist will give as a term of classification based on how many sequences you could actually see the tumor. If you could see the tumor on T2, contrast enhanced, diffusion weighted, and spectroscopy, if you could see the lesion on all of these cases, it's considered a high suspicion. And with the fusion system, then it's able to really increase the number of positive biopsy rates per patient compared to the standard of care. If we look at patients, what's interesting is that there are some cases that had prior negative biopsies. That means that they got some biopsies in the past that all came back negative, but their PSA was continually rising. So these patients actually came back, and if we look at those patients specifically, then we really see a jump in terms of moderate and high suspicion, especially in the high suspicion, we see a jump of around 22% in positive biopsy rate per patient by using this fusion system. And these are cases actually trying to analyze more in detail the targets that were missed. The target that was missed with the fusion system actually had a Gleason score, so that's a rate that's given with histopathology, had a Gleason score which was significantly lower than the ones that were missed on systematic biopsy. So the ones that were missed with the fusion system were actually not as high suspicion as the one that were missed on systematic. And here I'll show you just maybe a case study of a 63-year-old that had 4 prior negative systematic biopsies. So he came in 4 times. Every time he came in, he got 12 biopsies, so that's a total of 48 biopsies he got. And each time he came back negative. The problem is that his PSA was continuously rising. So a 5th time he came back, and this time the radiologist actually identified the suspicious lesions on the MRI. And what it probably explains why it was always negative is that the lesions were actually midline apical anterior central. That means that's a region of the prostate on top of the prostate that's fairly almost never sampled with transrectal ultrasound standard biopsies. So all systematic cores came back again negative, but with the fusion system, we were able to target all the suspicion lesions, and all came back positive with a Gleason score of 7 to 8. So really, that's one of those kind of showcase examples that shows that by really trying to focus on the lesions that were identified from the MRI, it's able to detect cancer, which was previously not be able to detect. So in closing, I would like to just show you a few of our other research that's using robotic surgery for image-guided intervention. So here with the Da Vinci system, try to use laparoscopic ultrasound fused with MRI. So for example, in the case of a nephrectomy, trying to remove necrotic tissue from a kidney. So we use a similar framework to try to co-register the laparoscopic ultrasound with MRI and confirm the regions that are actually going to be surgically removed with the Da Vinci system. And I talked about a bit about in-gantry MRI. So in collaboration with some partners in the University of Sherbrooke and a spin-off company from that, we're building a navigation system with all polymer MRI compatible robotic system that's able to be used inside of the gantry. So it's made of all polymer materials. So the actuators are pneumatic. And we're able to really have a needle guide that's completely compatible, even if we're using real-time MR imaging, will cause no interference during the actual navigation procedure. So we tested it on, right now really in the animal testing phase, but with an accuracy of 3 millimeters for some targets that were identified in the prostate of a beagle. So future directions, so I think really what we're driving to is to actually create better numerical modeling of the anatomy to have better prediction on how cardiac motion, respiratory motion affects image-guided interventions, and also use that for spatial temporal analysis, either for disease progression, but also to understand better the anatomy from a physiological and morphological perspective. So as I said, image-guided respiratory gain is a key issue that needs to be addressed to be able to, especially in abdominal interventions, where it really plays a key role, either for prostate or for liver also, be able to have a reliable statistical representation to compensate for these artifacts. Obviously, all the technologies we're developing needs to show some clinical utility. So a huge effort is made to actually show its benefit for decision-making, planning, or monitoring. And also developing more autonomous systems and more accessible systems, for example, low-cost ultrasound systems that could be used from non-experts, for example, in third world or emerging countries, to be able to use these systems in the hands of people that actually need them but don't have all the high-end expertise that we have here, and be able to integrate intelligent anatomy to localize some organs or some lesions that may not be detectable from the user's perspective. Thank you very much, and I'll be happy to answer your questions. That's from a single CT or MRI scan. Yeah.
You found that it is a problem start a procedure, is there any motion that you have to account for, particularly if you're going to apply motion with either the imaging ultrasound or even with a needle as you try and poke into the tumor?
Yeah.
How big of a problem does that
It will change during the procedure if before we do a registration, before doing the needle insertion, and we do a double check with the ultrasound, everything aligns properly. But once the needle is turned to be guided towards the tumor, it will, with time, it will change a bit the registration. So right now, what I showed for the liver, we didn't have a motion compensation algorithm, but it's definitely required to keep track of the motion and deformation of the liver as it's guided towards the tumor. The registration will also be affected based on where the tumor is. If it's a more peripheral tumor that's more in the periphery of the liver, the registration is based on the liver surface, so it will be very accurate towards these regions. If it's more of a deep tumor that's fairly far away from the skin entry point, then the accuracy of the registration will be different towards near the tumor. So depending on where the tumor is and how much insertion the needle has to be, it will affect kind of the registration process.
I also was wondering why you are stuck with an 8 millimeter slice spacing. What prevents you from improving that?
We just took the images that were standard protocol from the PAC system. So with our radiologists, we didn't develop a particular sequence for the liver segmentation from MRI. We just that was actually the based on the contrast agent, the specific sequence we needed. That was the kind of the best imaging that we had. But obviously, it could be improved.
I don't want to have
yeah. Well, the therapy will be kind of different. Probably, I'm not sure it will be really an ablation. If it's more of a diffuser, for example, for liver cancer, RFA will probably not be the best therapeutic solution for that. Patient will probably go through radiotherapy. If it's more diffuse procedures, or if it's an HCC, then it's more diffuse. They'll go through chemoembolization. And in that case, then yes, we're starting to work on that type of procedure. I think I had one slide on that. It was fairly localized. It's not as diffuse as. So this one is not fairly diffuse, but this is a primary liver cancer. The segmentation problem will be different, definitely. We need to adapt the segmentation algorithm to be able to process more diffuse tumors. Sometimes you'll have also multiple tumors that kind of collide together. So it's definitely a different challenge than trying to treat focalized tumors like metastases. In terms of registration problem, I think depending on what you use in terms of the structure, it could be fairly similar. But yeah, the therapeutical approach will be different, and the segmentation of the tumor will also be different.
So, with regards to the prostate stuff, so the MR variants are getting pretty good at identifying where suspicious lesions are. Do you think they'll ever get to the point where we won't need to use the biopsy anymore? Or what is the rationale you see for having to still take the biopsy if we're guiding it with the biopsy?
Well, I think they still need to confirm that with histopathology, being able to take tissue from the prostate and confirm that with pathology to be able that they have cancerous cells. I'm not sure they're up to the point where they could really treat just without any confirmation. I think we'll be able to target even better with better imaging and better sequences. But I don't know from an ethical point of view if that's doable, that you could treat just by seeing the tumor and not being able to really to confirm that by any more testing with the pathology to see if there's cancer cells. Maybe one day, but I don't see that really in the next few years.
I had one more question, if I may. Your segmentation method, is is that fair to say that it is a modification of the level set algorithm, or is this completely...
It's different. It's more inspired by the formal models, so more of the point distribution models. If you're talking for the MR segmentation, where we're trying to build training set of and using statistical knowledge and being able to deform a mesh by searching for optimal boundaries. It's more inspired from PDMs by COS.
If there are no other questions, I invite you to meet with Samuel Kadoury. Thank you very much.
Thank you.